Now some more basic concept about a typical antibody molecule, right? So we were talking about that antibody molecule, the tetrapeptide. Now we have to develop a relationship between tetrapeptide. We should be clear about these terms, and heavy chains, light chains, fragment FAB, fragment C, and then we should develop a concept what is monomer, monomer molecule, dimeric molecule, dimeric antibody molecules, pentameric, I will teach you this, pentameric molecules, what is the concept of affinity and avidity, right? And what is the concept of valency? You already know about the tetrapeptide. We said that every antibody molecule is basically a tetrapeptide made of 2 heavy chains and 2 light chains, right? And a typical molecule has 2 FAB fragments if it is treated by papain and 1 FC fragment. These terms I will explain now. We go back to our typical molecule, that these were heavy chains and these are light chains. All of them are held together by disulfide bond. Now actually this whole tetrapeptide is considered one monomer. This is considered one monomer, and in simple diagrams it is shown like this. Actually these part of the Y are representing FAB fragment and this stem of the Y is representing FC fragment. That is why very commonly you find antibody molecule like this, right, made in the books. So basically when you see an antibody molecule like this, it is fundamentally a tetrapeptide, but when these 4 peptides are held together by disulfide bond, we say this antibody molecule is one monomer. It is monomer. Is that right? Now sometimes it happens that more than one antibody molecules are held together, they are held together, this is one monomer, right, and this is suppose another monomer, and if these monomers are held together by another peptide chain, this peptide chain is holding them together, this peptide chain is joining them. So we call it joining component or J component. What we call it? J component. If more than one antibody monomers are held together by J chain or J component, right, or J chain, joining chain, we say now we are having multimeric molecule. Now we are having multimeric molecule, and this multimeric molecule has 2 monomers, so we call it, this multimer is dimer. This multimer is dimer. Sometimes it happens that with the joining component there are one, 2, 3, 4 and 5. Now in this case this J component is holding how many chains together? It is holding 5 chains together. So we call it pentamer. This is this type of you can say grouping of the antibody molecules, right, in which 5 monomers are together and held together by the J chain. This is called pentamer. So when we are talking about these terms, when I say that there is this antibody is monomeric, it means this antibody exists in this form. If I say there is another antibody which exists in dimeric form, it means it exists in this form. And if I say that a particular antibody group is pentameric, it means it is existing in this form. Now we have to develop the concept of valency also. Valency means that one group of antibodies can bind with how many antigens. For example, this can bind with how many antigens? Only with 2. So we say this monomeric molecule is having two valencies. So we say this monomeric molecule is divalent, and this monomeric molecule is tetravalent, and this monomeric molecule is, yes, it can bind how many antigens? 10. So it should be decavalent. You understand it? So now you should know that when we are talking about antibody molecules, these special terms are meaning exactly what concept. When we say an antibody molecule is tetrapeptide, it means a typical antibody molecule has 2 heavy chains and 2 light chains. When I say that antibodies of different classes may exist as monomer or dimers or pentamers, it means we are talking about that. If I say that, for example, immunoglobulin IgG, it exists in monomeric form, it means there is no J chain holding multiple molecules together. If I say that immunoglobulin A group in the body secretions are dimeric, it means they are held together by J chain, 2 monomers are held together by. Or if I say that immunoglobulin class of Igm in the serum is in pentameric form, it means it is holding how many? 5. But if I say that, let's suppose this molecule belongs to immunoglobulin G class, so we say this is monomer with divalency, and if it is IgA, this is dimer with tetravalency. If it is Igm molecule in the blood or plasma or serum, then it is pentameric molecule with decavalency. Decavalency. Am I clear? Any confusion up to this? There is no confusion? Okay. So this concept is clear, that valency refer to the group of antibodies binding to how many groups of antigens. Is that right? Or one antibody can bind with 2 antigens. It is divalent. But if it is dimer, it will have tetravalency. If it is pentamer, it will have decavalency. No problem. Now we have to come across another term, concept of affinity and avidity. Concept of affinity and avidity. Let me tell you that what is the meaning of affinity between the antigen and antibody. Right? Affinity. Let's suppose this is one antibody molecule. This is another antibody molecule. Now both of them, okay, first I will make the antigen here. This is the antigen, and here this is the antigen. Now this antibody molecule is having these variable, what are these? Variable regions which are binding with antigen. Now we make another antibody molecule, and this is what? Antigens. These are its which portion? Let's suppose this is antibody molecule number 1, this is antibody molecule number 2. What do you think? Antibody molecule number 1 is holding the antigen more tightly, or antibody molecule number 2 is holding the antigen more tightly? 2. Actually, these interactions between the variable and hypervariable region of antibodies and between the antigens, these interactions, they determine how tightly antibody will hold the antigen, right? This tightness or strength, the strength or the power with which one antigen binding site binds the antigen, this strength is called affinity. So molecule number 1 will have less affinity for the antigen, and molecule number 2 will have more affinity for the antigen. Is that right? For example, there is a beautiful girl here and her sister is holding her hand. Affinity will be more or less as compared to her boyfriend, a very new boyfriend holding her hand. Where affinity will be more? New boyfriend, not old one. Alright? So what I'm saying that affinity mean that if antigen antibody are interacting, how tightly they are holding each other together, or how what is the strength of the power with which one antigen binding site is interacting with the antigen, or as Shan is saying, exactly what is the binding force, what is the binding force between the antibody molecule and antigen. Am I clear? This concept is called affinity. But avidity is a slightly different concept. Affinity refer to the binding force between antigen and antibody at one point. Avidity mean a different thing, let me tell you. Let's suppose that there is a very big antigen. It's a very big antigen with multiple points with which antibodies can, yes, it has, okay, I will make it a simpler diagram so that you don't become afraid of this. Okay. This is a very big antigen with small epitopes. Epitopes are the points exactly with which the antigen binds. Now look, if a monomeric antibody come, this will react here and here only at 2 points. And if polymeric or multimeric antibody come, the J chain. Do you think monomer has only 2 points to bind, but dimer has how many points to bind? 4. And if pentamer is there, how many points of binding? Especially in large antigens, 10. So total force with which the, okay, another, I will repeat it again. This is monomer, this is dimer. Monomer is actually divalent and dimer is actually tetravalent, right? Affinity depend on, when we talk about affinity, we are just talking about that one antigen point and one antibody point interaction. But when we talk about avidity, we are actually looking at all the affinities in a group together. This binding force plus this binding force plus this binding force plus this binding force. Affinity at 1, affinity at 2, affinity at 3 and affinity at 4. All affinities together are called avidity. What are they called? Avidity. Is that right? So again I will repeat. Avidity mean that at how many points antigen antibody are interacting. As Igm molecules, they are pentameric, so their avidity is more. But IgG molecules are only monomer, so they are having only divalent, so their avidity is less. Am I clear or not? No problem up to this? Okay. Now we develop some concept about the different classes of antibodies. So antibodies have, all of you know that basically antibodies are having 5 main classes. Now according to which concept, what is the reason to divide your antibodies into 5 classes primarily? You have any idea? Why antibodies are primarily divided into 5 classes? There must be a reason. Yeah?
Maybe their functions?
No, that is not the real reason. Why antibodies are, for example, look, you bring from my blood, again, from my blood you bring all the plasma protein out and run the plasma proteins on the electrophoretic strip, and from the gamma band you bring all my antibodies out. All the antibodies which you bring out of my gamma component of the plasma proteins, they can be basically classified into 5 groups. On what base they are classified? Yes?
Is it the structure, something to do with the heavy chain?
Yeah, that's good, that's good, very good. Actually, primarily 5 types of heavy chains can be produced and 2 types of the light chains can be produced. The plasma cell can produce primarily how many types of heavy chain? 5 types of heavy chains and 2 types of light chains. Now, if you if you put all my antibodies out of my bodies, you bring all my antibodies out and classify them according to the heavy chains, how many groups will be found? 5 groups. Let's suppose this is one group of heavy chain, this is another type of, okay, I will make it a diagram and then I will explain that why I am making it like this. These are just, I am drawing the heavy chains. How many types of mainly heavy chains our B cells can produce? Mainly 5 types, even though there are subgroups also. We'll talk about that. 1, 2, 3, this is the 4th and 5th group. Right, so these are the types of heavy chains which can be produced, and the light chains which can be produced are only 2, the kappa and lambda. Now, antibodies are classified according to the heavy chains into how many classes? 5 classes, because mainly 5 types of heavy chains can be produced. So if you put all my antibodies in the table and if you can look at their FC portions are made of heavy chains. FC portions are made of heavy chains. How many different types of heavy chains you will find? Primarily 5. Is that right? So we'll say they are basically 5 types of heavy chains and 5 classes of immunoglobulins primarily. Is that right? But light chains are only of 2 types, kappa and lambda. Heavy chains are like, suppose this heavy chain is called gamma chain, this heavy chain is called alpha chain, suppose this heavy chain is called mu chain, this heavy chain is called delta chain, and suppose this heavy chain is called epsilon chain. This heavy chain is called epsilon chain. So basically antibodies can be classified depending upon the type of heavy chains into 5 classes. But if you look at their light chains, there are only 2 types of light chains. There are kappa light chains, suppose this is kappa light chain, and another is lambda light chains. Actually, if you remove all the antibodies with the gamma heavy chains, some of them will be having lambda light chains, of course held together by disulfide bond, and some of them will be having lambda heavy chains, light chains. Is that right? So actually kappa and lambda light chains are present in every group. So we can say some of the molecules in every group will be having, what is this? Kappa light chains, and some of the molecules will be having lambda light chains. Is that right? And of course you are so intelligent, all of them are held together by disulfide bonds. There is no need to make it, you understand it. It means if you have these 5 group of antibodies or 5 classes of antibodies, why the antibodies in different classes are different? Because they have different heavy chains. And why antibodies in different classes have some similarity? Because they have same light chains. Again listen, we say that antibodies are basically having 5 classes. Why we divide the antibodies into 5 classes? That classification is depending upon the types of heavy chains. Is that right? So in one group, in any one group, suppose we talk about this group, if we take antibodies of only this group, all the heavy chains will be same. Is that right? But some light chains will be kappa and some light chains will be lambda. So when you study in comparison group to group, why groups are different from each other? Because they are having different heavy chains, but they do have some physico-chemical characteristics which are similar, and they are due to light chains. Am I clear? No problem up to this? Now, those antibodies which are, those immunoglobulins which have gamma heavy chain, they are called IgG. Those immunoglobulins which have alpha heavy chain, they are called IgA. A. And those immunoglobulins which have gamma heavy chain, they are called Ig G. Immunoglobulin G. Sorry, this is mu. So they are called IgM. And those immunoglobulin molecules which are delta chain, delta heavy chain, they are called IgD. And those immunoglobulins which are having epsilon heavy chain, they are called IgE. That is why we say there are 5 classes of the immunoglobulins, because there are 5 different types of primarily heavy chains. Then later on you will learn that there are 2 subclasses of IgA, that is IgA1 and IgA2. And in the same way IgG can be divided into 4 types: immunoglobulin G1, immunoglobulin G2, immunoglobulin G3 and immunoglobulin G4. Is that right? Any problem up to this? So these are the subclasses. IgG has 4 subclasses, IgA has 2 subclasses. No problem up to this? Now let's go into detail of every class one by one. We'll start studying now in detail every subgroup. First of all, we'll discuss about IgM. Important points related with immunoglobulin IgM. Immunoglobulin IgM. Of course, you will tell me that IgM has what type of heavy chain? Mu type of heavy chain. Now another question which I want to make it clear. Before I really go into detail of classes, let's suppose you have a bacteria, and on this bacteria, one bacteria has only one type of antigen, or one bacteria may have many types of antigens. One bacteria may have one type of antigen, or one bacteria may have many different types of antigens. Yeah. One bacteria, suppose this is one bacteria, it may have star antigen, it may have ball antigen, it may have triangular antigen, it may have square antigen. Actually, one microbe may have different types of antigens. That is why we say microbes are antigenically mosaic. Right? Every microbe has multiple type of antigens. Now, of course, the antibody which is made against a microbe, that will be one type of antibodies or multiple type of antibodies against. For example, this bacteria enter into your blood or in your body. Suppose this is E. coli. Okay, this is spherical, so it cannot be E. coli. It is Staphylococcus aureus. It enters into your body, and you build an immune response. Right? This immune response will have antibody directed against only one antigen, or antibodies will be produced which will be against multiple antigens. Multiple antigens. So there will be anti-star antibody, there will be anti-gola antibody or anti-ball antibody, maybe anti-triangular antibody, anti-square antibodies. What I am saying, that against a microbe which has multiple antigens, it is quite possible that you will build immune response against multiple antigens. This type of immune response is called polyclonal immune response. One clone of plasma cell or B cell will produce anti-star antibodies. Another clone of B cell or group of B cell will produce anti-ball antibodies. Another group of B cells will produce anti-triangular antibodies or anti-square antibodies. Am I clear? So usually against an organism, the antibody response is polyclonal. Right? That microbes are antigenically mosaic, and immune response from a healthy person, immunocompetent person, is polyclonal immune response. Now another thing, let's suppose we talk only about anti-star antibody. Let's suppose this star antigen come into your body and we are making anti-star antibody. This anti-star antibody will belong to which class? It will belong to IgG or IgM or IgA or which class? It can belong to any group. Against one antigen, we may make IgM in the beginning, and later on we can make IgG or even IgA. This is very important concept, extremely important. Against the same antigen, at different times or simultaneously, you may be producing antibodies of different classes. Now let me make this concept more clear to you. Let's suppose this is star antigen. Now these are suppose heavy chains are suppose mu heavy chain. Oh, okay, these are gamma heavy chains. If these are gamma heavy chains, then can you tell me which is the antibody which is reacting with the star? Which class of the antibody is reacting with the star? If it is gamma, Ig, G. This is IgG molecule because it is having a heavy chain belonging to gamma. But the interesting thing is that now heavy chain has been changed, but specificity for the antigen has not changed. Is that right? And now let's suppose it is mu. If this heavy chain is mu, then what is this antibody? Class IgM. Let's suppose that this heavy chain is, yes, alpha. If these are alpha heavy chains, then what is this? IgA. You see, against the same antigen, you can produce IgM, IgG, IgA. Are you understanding me? Right? So against one antigen, you can produce antibodies with different classes. Usually the first class of antibody which is produced is IgM, and later on you produce IgG against the microbes. When microbe first time enter into your body, in the antibodies you use which heavy chain? Mu. Usually when antigen is entering in your body first time, when plasma cells are making antibody on first exposure, they use which heavy chain? Mu. Because they are using the mu heavy chain, so antibody class which is initially synthesized against antibody is IgM. But later on class switching occur. The same plasma cell may start producing IgG. Now you may be thinking how it is possible. Have you ever thought of it? How this class switching occur? How one class of antibody switch to another? Let me explain very briefly. Later on we will have a full lecture on class switching and genetic. Suppose this is a plasma cell, right? This is the nucleus of the plasma cell. Let's suppose here are the genes for the heavy chain. You know, in the heavy chain, this is a portion, this will make which? Mu. When this part of the genetic material expresses itself, you make which chain? Which heavy chain you make? Mu. And when this start expressing itself, you make which heavy chain? Delta. And when next gene, let's suppose this gene start expressing itself, what is this? Gamma. So this is the gene for mu, this is the gene for delta, this is the gene for gamma, and this is the gene for alpha, and this is the gene for epsilon. Is that right? Now these are the genes which are present in every plasma cell to synthesize a special type of what? Heavy chain. Is that right? Now let's suppose this is a special part of gene, and this part of the gene is concerned with the heavy chain variable, and this is heavy chain constant for the mu, this is the heavy chain constant for the delta, this is the heavy chain constant for the gamma, this gene is for heavy chain constant for alpha, and this gene is for heavy chain constant for epsilon. Now you listen very carefully. When first time antigen come, right, this part of the DNA will combine with the mu. So antibody which will be formed, that will be, suppose this part of the DNA makes the variable portion of constant which will react with star. Now this is, suppose, make the peptide in the heavy chain which will react with, bind with the star antigen. Is that right? This is the genetic component of the B cell or plasma cell, that when it translates into a peptide fragment, that piece of the peptide will become the variable portion of the heavy chain which will react with a special type of antigen which is, suppose, star antigen. Now what really happens? Initially this fragment fuses with this and both of them come together and start making heavy chains. So in the beginning the heavy chains which are made in this cell, they will have this portion with this portion. And what is this? This will be actually, if I really put it like that, what is this? This is a heavy chain for the Igm. Is that right? So in the beginning, this cell will be making heavy chains for the Igm. And the antibodies which will be produced, of course, what will happen? This heavy chain with another mu heavy chain with two, let's suppose, kappa, and what will this become? Whole molecule will become Igm molecule and it will be secreted out. But if antigen again and again come and exposure become prolonged or repeated, this will be digested away and thrown out. It will change its strategy. And this portion will fuse with this one. Both of them are thrown out. Both of them are thrown out, loop out and destroyed. Now the combination will be made VH with gamma. Now the heavy chain which is made now, this is having this, this gene and this gene fragments will come together. And new combination will make the peptide chains, right, which will still bind with the star. So bind with the same antigen. But of course these are the light chains. But heavy chain has been changed. The constants of the heavy chain has been changed. And now class of the antibody has switched from Igm to IgG. You are understanding? So against the same antigen, plasma cells are initially producing Igm and later on they switch the class and start producing IgG. So against the same bacteria and against the same antigen, initially you may produce Igm and later on you may produce IgG or you may produce IgA. Am I clear? No problem into this? Is that right? So this is the concept of class switching. Because the this portion of antibody is different from class to class, right? But this portion of antibody may be same for a given antigen, even classes may be different. Am I clear? And due to this reason, whenever you get recent infection in your blood against that infectious agent, Igm level will go up. But if infection is repeatedly there, then IgG levels will also go up. So if you are having some disease, right, and I check your blood and against an antigen you have only Igm, it means you had recent infection. But against another disease, I check your blood and I find IgG but I do not find Igm. It means there was some previous infection. Are you understanding? And if I find against a bacteria IgG and Igm, Igm means infection is going on. IgG means it's quite long time. You are understanding it? So it's prolonged infection. Again, let me tell you something. If this is my friend, this marker is my friend, against a bacteria it has only Igm. It means it got bacteria recently. This is my friend, against the same bacteria it has only IgG. Probably it has previous infection. This is my friend, against the same bacteria it has Igm and IgG both. It means he has infection from long time, that is why he has IgG and still it is present in his body and he is still making Igm. Am I clear?
Could it also mean that he had the infection before and then he recovered and he is getting a recurrent?
It is quite possible. It is quite possible someone who has against a specific antigen IgG and IgM, maybe IgG represents the past infection and IgM represents recent infection. Am I right? No problem up to this. Here, now we go to details of the antibody classes. Now, we will go to the IgM molecules. First of all, we will discuss IgM class of the antibodies or immunoglobulins. The very important thing that IgM is normally expressed on the membranes of the B lymphocytes. This is the B lymphocytes, and on the surface of the B lymphocytes, IgM molecules are expressed. What is the purpose of expressing the monomers of IgM on the surface of B cells, B lymphocytes? Yes, I will be impressed if you can tell me. Okay, let me tell you before you tell me something new. Suppose this is one B cell, this is another B cell, this is another B cell, right? Let us suppose this B cell is presenting IgM on its surface, and this IgM can bind only with the ball antigen, right? This B cell is presenting IgM on the surface, and this IgM can bind with the star antigen. This is another B cell, and this B cell has IgM molecules which could bind with square antigen. Actually, every B cell in your body expresses IgM molecules on their surface and different IgM molecules on the surface of the different B cells bind with different antigens. Let's suppose these three B cells are there and there is no antigen, and these three B cells are there in your body. If star antigen come, this will be activated. If ball antigen come, only this will be activated. If square antigen come, only that will be activated. So actually, there are millions and millions of the B cells having special type of IgM on their surface, and these IgMs are going to act, these IgM surface IgM molecules are going to act as a receptor for the receiving molecule, receptor for the antigen. That is why these surface IgM molecules are called BCRs, B cell receptors for the antigens. So next time someone say that B cells are having receptors for the antigens, most of these receptors for the antigens are actually IgM molecule. Some of these may be IgD molecule. So IgM mainly, and sometimes IgD also act as B cell receptors, right? So actually different B cells have a capability to produce different type of, different B cells have a capability to produce antibody for which are specific for different antigens. All B cells cannot make antibodies for same antigen. Is that right? So actually every B cell on its head is having a sample antibody, having a sample antibody. So actually antigen goes and bind with the relevant, and then that B cell will be activated and convert into plasma cell. Now if star antigen come, only this B cell will convert into plasma cell. But if square antigen come, do you think this will be activated? No, that will be activated. So right, so what really happens that antigens come and bind with only that B cell which is having such IgM or IgD molecule which can react with specific type of antigen. So we can say one function of the IgM molecule is that they act as antigen receptor on the membrane of B cell, and their IgM molecule is present as monomer. So these are acting as BCR. So this is one important action of IgM, right? But IgM is not only present on the surface of the B cells, which is called surface IgM or membrane IgM or B cell IgM, they are monomeric. But actually IgM molecules are actively secreted by plasma cells, and IgM molecules are present also in your blood, in your serum, and serum IgM is pentameric. They are not monomeric, right? Suppose this is your blood, this is your circulation. In your circulation, let's suppose this star is the J chain. Then how many IgM will be there? 1, 2, 3, 4 and 5, right? So basically IgM molecules are present in our serum or plasma as pentameric molecule, which are joined together by the J chain. Is that right? Because they are large molecule, they are mostly present in the blood, they are not present in interstitial fluid, and they are not part of other body fluids. IgM is mostly distributed in plasma. Is that right? This is the another thing, that IgM, here it is pentameric, but it is decavalent. I told you IgM is the first antibody which is produced during the immune response to a new antigen, right? Such immune response is called primary immune response. When first time you get antigen and you develop antibody, we say it is primary immune response. But if antigen is coming repeatedly, then you are developing more immune responses. After the first immune response, whatever later on immune responses come, they are called secondary immune responses. Let me tell you here that let's suppose you are vaccinating a person, you are giving vaccination. This is first vaccination, second vaccination dose, third dose. During the first dose, you will develop gradually IgM, but then IgM level will go down. But with every dose, it may go up. But during the first response, the IgG will be very less. In second response, IgG will become very high. Third response, it will become very high. Now listen. This, when antigen first time enter into your body, you develop an antibody response after some delay. After some delay, from here to here there was no antibody, gradually it started. This is called lag period. What is it called? Lag period. So when first time antigen comes, there is a long lag period, and then antibodies will start appearing in the blood very slowly, is that right? And they will go a very low level of peak, and then gradually they will come down. This immune response is called primary immune response. And in the primary immune response, main antibody is IgM, and IgG and other antibodies are very less. But if you repeat the, okay, I'll make it more clear. Let's suppose first dose, second dose, third dose of vaccination. First response is right, this is IgM. So you see, with every time antigen comes, there's a little bit IgM response. Is that right? And initially this is first response, but IgG response, this is very little in the beginning. Second time it is very rapidly going up, goes to a peak, stay for a long time, come down, but not so much, and then next dose, third dose, it goes very high and stay for very long high. So actually we say with repeated antigen exposure, IgM is not boosted, but IgG is boosted. Is that right? So when you give me star antigen first time, I will have mostly anti-star IgM, but very little anti-star IgG. But after a few months you repeat star antigen, then I will have still little IgM, but lot of anti-star IgG. And this time it will come very rapidly, it will go very high level, and it will come down and stay on the high level, right? This type of response is called secondary response, but this type of response is primary response. So on first exposure of the antigen, antibody level go up slowly, goes to a very low peak and gradually come down and persist at very low level. But on repeat exposure, antibody level goes very rapidly very high, achieves very high level, stays there for longer time and come down slowly. Right? On every repeated exposure, this will behave very briskly, antibody level go up and stay very high for longer time. This first response is called primary response, the second response is called secondary response, and this third response is called, yes Dr. Safa? It is not called tertiary. First response is primary response, after that every repeat exposure is secondary response. Characteristics of primary response are long lag period, very few antibodies formed, and it comes down and stay very low. This is primary response, and main antibody is IgM. But in secondary response, what happen? Yes. There is short lag period, very rapidly rising antibody level, very high peak is achieved, which stays for longer time and come down, but stays at higher level. This type of pattern is called secondary response. Secondary response has main antibody of Igm class. Why it happens so? Look, when first time some plasma cells started, they were mainly producing IgM. You know, bone marrow is producing the B cell. After 6 months, when second dose comes, some new B cells have come, they again produce IgM. Then after 1 year again exposure occurs, then again some new come and IgM. But from where the IgG come? IgG come when these first IgM have done class switching. So next, these are called memory cell. So next time when antigen come, previous memory cell very rapidly bigger response. So these are the rapid response by the memory cell which were in the previous exposure IgM producing cells. Now they are producing IgG. When you come third response, then memory cell from here and memory cell from here, both of them respond. So every time when you are boosting, you are increasing the memory cell against the antigen, and these memory cells are having what? Capability to produce more and more IgG. That is why IgG keep on getting boosted with every response, but IgM does not get boosted. Am I clear? No problem up to this? Now we were talking about that, so we can say that IgM is surface IgM, which is acting as B cell receptor. Number 2, there is serum IgM, and about serum IgM what you should know? That it is pentameric, yes, pentameric. There are 5 monomers of IgM held together by J chain, and this is decavalent, because it can bind with how many antigens? 10. So it is having less avidity or very high avidity? It has very high avidity. Is that right? So because of another thing, because it is decavalent and it can react with multiple antigens, now you imagine, let's suppose this is IgM against your antigens on RBCs. If this is IgM pentamer against the antigen on RBCs, it will react with one RBC or multiple RBCs. And one RBC may be reacting with multiple pentamers. So what is happening? So what really happens that, are you understanding what is happening? The network RBCs which were originally separate from each other, as soon as IgM pentamers come, they cross link them and these RBCs clump together. This reaction is called agglutination. So IgM can react with multiple antigenic particulate matter, particles. So it agglutinates the particles. So it can agglutinate the RBCs. If there are multiple bacteria, let's suppose these are the bacteria, and you know every bacteria has multiple antigen or one antigen? Multiple antigen, and every, for example, this bacteria has 3 types of antigens and every antigen is multiple copies. So if there is IgM, so IgM will react against this, react against this, react against this. So all these are, what is happening to them? They are cross-linked. These bacteria are cross-linked. So we say bacteria are agglutinated. So we say that Igm antibody is agglutinating antibody. That if there is some antigens which are in particle forms, it will make the aggregates of particles, right? So number one, Igm is act as B cell receptor. Igm in the blood is or serum is pentameric with decavalent, and it is agglutinating. Yes, agglutinating what? Antibodies. These are agglutinating antibodies, and of course you will remember this is antibody of primary response or secondary response. Primary response. It's antibody. In primary response, Igm is the main antibodies. In secondary response, it is far less than IgG. Am I clear? Then another thing about the Igm is that Igm can activate complements, right? You know, complement proteins are produced by the liver, right? And Igm can activate the complements. There are two antibodies classes which can activate the complements. By that number one is IgG and number two is Igm, because Igm antibodies, these are 5 groups held together by J chain. Actually, these Fc portion of the Igm can bind the complement proteins with them, and they can activate complement. This is C1, and C1 can activate C4, C2, you know it. Then C3, C5, 6, 7, 8, 9. So this pathway of complement activation where antibody Fc portion can bind with C1, 4 and 2 and activate the complement pathway through this pathway, this is called classical complement pathway activation. What is this? Classical activation of the complements. So Igm has one more function. What is that? This is complement pathway activation. But there is one thing, the Fc portion, the receptors for Fc portion of Igm are not present on our cells. So Igm cannot bind with our cells. Let me tell you. If there is a bacteria here, how the Igm is going to kill? Igm can kill the bacteria or destroy the bacteria by activating the complements. Let's suppose here is Igm. Of course, now this group of Igm will activate the complements C1, 4, 2, C3, 5 and 6, 7, 8 and 9. You know C5b, 6, 7, 8, 9, this is called membrane attack complex. This you will study in detail in complement system activation. So C5b, 6, 7, 8, 9, this complex will puncture the membranes of the bacteria and bacteria will die, right? But Igm Fc portion cannot bind with neutrophils or macrophage cells. Why? Because the Fc portion does not have receptors on the phagocytic cells. So it cannot opsonize the bacteria. You know, opsonization is a different concept. Opsonization means that let's suppose here the bacteria, here is antibody molecule. Antibody molecule on one side bind with the bacteria, on other side antibody molecule has a receptor on a, this is a receptor for on a, suppose what is this cell? Suppose this is neutrophil of macrophage. So on the surface of neutrophil of macrophage, if there is a receptor for Fc portion of the antibody, and an antibody on one side catches the bacteria, on other side binds with the neutrophil of macrophage, can now this bacteria run away? It is caught, and then very effectively it will be phagocytosed. This bacteria cannot run away. Is that right? Because there is an antibody which can bind on one side with the bacteria and other side with the phagocytic cell. Right? And phagocytosis of this bacteria is facilitated in the presence of this antibody. Such antibodies are called opsonins. What are they called? Opsonin. Igm is not opsonin, because if Igm bind here, Igm cannot bind with the phagocytic cells. This is IgG. So antibodies belonging to Ig class act as opsonin, but antibody belonging to Igm class cannot act as opsonin. Is that right? So about the Igm, just important points. Number one, Igm is monomeric, right, and present on the B cell surface as B cell receptor. In the serum, Igm is present as pentameric, decavalent, large molecule, mostly in the plasma circulation, right, because they are trapped in that, not in the other body fluids in significant amount, and Igm is pentameric with decavalent molecules, and it can produce agglutination reactions. Is that right? It can activate the complement by the classical pathway, and of course we should remember Igm is the main antibody of the primary immune response. Is that right? Do you have any question up to this? So let's have a break.